Welcome back to the deep dive. Today, we're kind of zooming out. We've talked a lot about those tiny changes, point mutations in single genes.
Right, the small stuff.
Exactly.
Right.
Now we're looking at the big picture. Massive structural changes to the chromosomes themselves, chromosome mutations or aberrations.
Yeah, these are wholesale modifications. We're talking about changing the amount of genetic information or rearranging whole sections. Big stuff.
And the really key thing for you listening is that even though these changes are huge, structurally speaking, they actually follow predictable Mendelian inheritance patterns. It's not just chaos.
No, not at all. But it highlights this fundamental concept. Our genetic information exists in this really delicate balance. Our source material really hammers this home. Even small shifts in quantity or location can cause, well, pretty major phenotypic changes.
Often lethal, especially in animals, right?
Very often lethal, yes. So today, our mission is really to unpack how this architectural precision can break down. Where do we start? Probably with just counting errors.
Okay, yeah, let's start there. Changes in chromosome number. So when we talk about counting errors, there are two main flavors, right? Aneuploidy and euploidy.
That's it. Aneuploidy is probably the one people are more familiar with, maybe without knowing the term. It's when you gain or lose one or maybe a few chromosomes, but crucially, not a whole set.
Like having an extra book or missing a book from one volume of an encyclopedia set.
Exactly. The common ones are monosomy, that's two euplans and missing one chromosome, and trisomy, two plans plus euploid have an extra one.
Okay. And the other category, euploidy.
Euploidy means you have complete sets. Normal diploids, like us, are euploid. But if you have more than two complete sets, that's polyploidy. Triploid, $3, tetraploid, $4, and so on. More common in plants, but we'll get there.
So how do we end up with aneuploidy, that extra or missing chromosome? Where does that typically come from?
The usual culprit is something called nondisjunction. It's basically a mistake, a random error that happens during meiosis when sperm and eggs are being formed.
A sorting error, essentially.
Precisely. The paired homologous chromosomes are supposed to separate in meiosis the second, or the sister chromatids should separate in meiosis the second. Nondisjunction is when that separation fails.
So what happens then?
Well, you end up with gametes, sperm, or eggs that are abnormal. They might have two copies of a particular chromosome or none at all.
And if one of those fuses with a normal gamete during fertilization?
But the resulting zygote is either monosomic, missing one, or trisomic, has an extra. And that's the origin of conditions involving sex chromosomes, like Klinefelter, which is XXY, or Turner syndrome, which is just X.
Okay, let's talk consequences. Monosomy losing one chromosome. You mentioned it's often lethal, particularly for autosomes, the non-sex chromosomes. Why is losing one so much worse than gaining one? You'd think having one copy of each gene would be okay.
It seems counterintuitive, but it comes down to that genetic balance, that dosage. There are two main reasons it's so detrimental. First, losing a chromosome means you lose your backup copy. Any recessive lethal alleles on the remaining chromosome are immediately exposed.
No hiding them.
Right. There's no normal allele on a homolog to mask the effect. The second, and maybe even more critical reason, is something called haploinsufficiency.
Haploinsufficiency.
Yeah. It just means that a single copy of a gene might not produce enough of its protein product for the cell or organism to function properly. Think of it like trying to build something sturdy with only half the required materials. It often just doesn't work.
Although plants seem to handle it a bit better.
They do. Plants like maize or tobacco, they can tolerate monosomy better than animals, generally. They're often less vigorous than the normal diploids, but they survive. Plants just have more flexibility in their genetic makeup, it seems.
Okay, so what about trisomy? Gaining that extra chromosome, $2 plus $1 or one. You said it's generally tolerated better than monosomy.
Generally, yes, especially if the chromosome involved is relatively small, carrying fewer genes. But better tolerated doesn't mean good. It still usually causes significant problems. However, there's one major exception in humans for autosomal trisomies.
And that brings us to Down syndrome, trisomy 21.
Exactly. It's the only human autosomal trisomy where individuals commonly survive well into adulthood. It occurs in about 1 out of every 800 live births.
And the characteristics, the facial features, the developmental delays, they're linked to having that extra copy of chromosome 21.
That's the idea. Researchers have pinpointed a specific region on chromosome 21 called the Down syndrome critical region, or DSCR. The hypothesis is that having 3 copies of the genes in this region leads to an overexpression, too much product, which causes the specific phenotypes associated with the syndrome. It's a dosage issue again.
And where does that extra chromosome usually come from?
Overwhelmingly, about 95% of the time, the extra chromosome 21 originates from the egg, meaning the nondisjunction event happened during eugenesis in the mother.
Which leads directly to that really striking connection with maternal age.
It's traumatic, yeah.
Yeah.
The risk of having a child with Down syndrome stays relatively low for mothers in their 20s, but then it starts to climb sharply.
Yeah.
By age 35, it's significantly higher, and by age 40, the risk is about 1 in 100.
Why such a strong link?
It's thought to be related to the timeline of egg development. Oocytes actually begin meiosis when the female is still a fetus, but then they pause in meiosis the first. They stay in that suspended state for years, decades even, until ovulation.
Wow, decades paused in meiosis.
Yeah. And the thinking is that the longer they stay in that arrested state, the greater the chance that something goes wrong with the chromosome segregation machinery when meiosis finally resumes. Hence, the increased risk of nondisjunction with age.
It really underscores how precise meiosis needs to be.
Absolutely. And consider this. Studies looking at spontaneously aborted fetuses show that maybe 20 to 30% have some kind of chromosome imbalance. It just shows that having the correct diploid number is incredibly important for normal development and survival.
Okay, so we've covered errors involving one or two chromosomes aneuploidy. Let's shift gears to euploidy, specifically polyploidy, where we're dealing with entire sets of chromosomes. You said this is rarer in animals, but common in plants.
Much more common in plants, yes. Though it does occur in some lizards, amphibians, and fish, we usually classify polyploidy based on where the extra chromosome sets came from.
Okay.
First, there's autopolyploidy. This is when you have multiple sets of chromosomes, but they all originated from the same species. So a diploid, AA, might give rise to a tetraploid, AA, for example.
How does that happen naturally?
Usually through a failure in meiosis where all chromosomes fail to segregate, leading to a diploid gamete, $2. If that fuses with a normal haploid gamete, you get a triploid, $3. Or if two diploid gametes fuse, you get a tetraploid, $4.
And we can induce this too, right? I remember reading about colchicine.
Yes, exactly. Colchicine is a chemical that disrupts spindle fiber formation during mitosis. So chromosomes replicate, but the cell doesn't divide. You effectively double the chromosome number within a cell.
Why would we want to do that? What's the use?
Agriculturally, it can be very useful. Polyploid cells are often larger, which can lead to larger plants or fruits. Also, odd-numbered polyploids, like triploids, 3-toners, are usually sterile because their chromosomes can't segregate evenly during meiosis to make balanced gametes.
Ah, which is perfect for
Seedless fruits, like commercial bananas, which are triploid and propagated asexually. Seedless watermelons, wine sap apples too. And some polyploids just have enhanced traits, like the large size of the commercial octoploid, eaten strawberry.
Okay, so that's autopolyploidy, multiples from the same species. What's the other type?
That's allopolyploidy. This involves combining chromosome sets from different, though usually related species.
So like a hybrid situation.
Exactly. You start by hybridizing two different species, let's say species A and species B. The hybrid offspring, AB, gets one set of chromosomes from each parent. Usually these hybrids are sterile because the A and B chromosomes aren't homologous and can't pair properly in meiosis.
Right.
But if a spontaneous doubling event occurs in that hybrid, maybe similar to how colchicine works, you can end up with an ABB organism. Now, every A chromosome has a homologous A partner, and every B has a B partner.
And that makes it fertile.
Yes. This AABB type is called an amphidiploid. It's fertile because it can now undergo proper meiosis. This process is thought to have been really important in plant evolution.
Are there common examples of this?
Oh, definitely. Cultivated American cotton is a classic example derived from crossing an Old World strain with a wild American species, followed by doubling. And a really important man-made example is triticale.
Triticale.
Yeah, it's a hybrid between wheat, genus Triticum, and rye, genus Sicali. Scientists created it to combine the high yield and protein quality of wheat with the ruggedness and disease resistance of rye. It's an amphidiploid.
Fascinating. Okay, before we move on from number changes, briefly, what's endopolyploidy?
Ah, right. Endopolyploidy is just when certain cells or tissues within an otherwise diploid organism become polyploid. It doesn't affect the whole organism. For instance, some human liver cells can be tetraploid, 4 times on 4, or even octoploid, 8 times.
Why would just some cells do that?
The thinking is it might allow those specific cells to produce much higher levels of certain gene products they need in large quantities. More chromosome sets mean more gene copies to transcribe.
Okay, got it. So we've covered number variations. Let's move to the second major category. Changes in the structure of chromosomes. Deletions, duplications, and rearrangements.
Right. These usually happen because of breaks occurring somewhere along the chromosome. The cell's repair machinery tries to patch things up, but sometimes it reconnects the pieces incorrectly.
And if an individual inherits one normal chromosome and one that's structurally rearranged, making them heterozygous for the aberration, meiosis gets really tricky, doesn't it?
It really does. Let's take them one by one. First, deletion. Simple enough, a piece of the chromosome is lost. It can be from the end, terminal, or from the middle, intercalary.
Is there a well-known human example?
Yes, Cre-Du-Chat syndrome. This results from a deletion on the short arm of chromosome 5. Affected infants have this distinctive high-pitched cry, like a cat, hence the name, along with intellectual disability and other physical anomalies. The missing genes on that segment are critical.
Okay, so deletion is loss. What about duplication?
Duplication is the opposite. A segment of a chromosome is repeated, so you have extra copies of the genes in that region.
Is there a classic example here too?
For duplication, the textbook example comes from Drosophila, the fruit fly. There's a phenotype called bar eye, where the fly's compound eye is much narrower, like a bar. For a long time, it was thought to be a standard gene mutation.
But it wasn't.
No. Careful work by Bridges and Muller showed it was actually caused by a duplication of a specific region, 16A, on the X chromosome. Flies with more copies of this region had even narrower eyes. It was one of the first links between a specific phenotype and a visible chromosomal change.
Okay, now this is where it gets really interesting from an evolutionary standpoint, right? Duplications.
Hugely important. Susumu Ono proposed back in 1970 that gene duplication is perhaps the major driving force behind the origin of new genes.
How does that work? It seems like just having extra copies wouldn't create something new.
Well, think about it. If you have two copies of an essential gene, one copy can continue performing the original vital function. The organism stays alive.
Okay.
But that second copy is now somewhat redundant. It's free, evolutionarily speaking, to accumulate mutations without causing immediate harm. Over time, these mutations might cause it to diverge and take on a completely new, related, or even unrelated function.
I see. The original keeps the lights on while the duplicate can experiment.
Precisely. It's thought to be the origin of gene families, like the different globin genes that carry oxygen, or digestive enzymes like trypsin and chymotrypsin. Even genes implicated in human brain evolution, like the SRGAP2 family, seem to have arisen through duplication events.
And this isn't just ancient history, right? These duplications and deletions happen in humans now?
Absolutely. We now talk about copy number variations, or CNVs. These are relatively large duplicated or deleted segments, sometimes spanning many genes. They're surprisingly common in the human genome. And while many have no effect, others are increasingly being linked to susceptibility to complex conditions like autism, schizophrenia, certain cancers, and autoimmune diseases like Crohn's.
Wow. Okay, so we have deletions and duplications. What about rearrangements? Let's talk inversions.
An inversion is when a segment of a chromosome gets flipped 180 degrees. So the order of genes in that section is reversed. Importantly, there's usually no loss or gain of genetic information, just a rearrangement.
Are there different types?
Yeah, the main distinction is whether the inverted segment includes the centromere. If the centromere is outside the inversion, it's paracentric. If the centromere is within the inverted segment, it's pericentric.
Why does that distinction matter?
It affects what happens during meiosis. If the individual is heterozygous, for the inversion having one normal and one inverted chromosome. For those chromosomes to pair up properly, synapsis, they had to form this characteristic inversion loop.
A loop. Okay.
And if a crossover event, genetic recombination happens within that loop, things get messy.
How messy?
Very messy, especially with paracentric inversions. Crossovers there can produce chromatids that are dicentric, two centromeres, or acentric, no centromere. These are typically unstable and get lost during cell division, or they break apart. The resulting gametes are often inviable.
So it leads to reduced fertility.
Often, yes.
Yeah.
But inversions also have an interesting evolutionary consequence. Because crossing over within the loop leads to non-viable products, inversions effectively suppress the recovery of recombinant chromosomes for the genes within the inverted segment.
Why is that good?
It means that a particularly advantageous combination of alleles within that inverted segment can be kept together, inherited as a block, generation after generation, without being broken up by recombination. It preserves adaptive gene complexes.
Clever. Okay, last major structural change. Translocations.
Translocations involve the movement of a chromosomal segment to a new location in the genome. The most common type is a reciprocal translocation, where two non-homologous chromosomes exchange segments. It's like they swap pieces.
So again, no net loss or gain of material, just moved around.
In a balanced reciprocal translocation, yes, the total amount of genetic material is normal. But just like with inversions, meiosis in a heterozygote is complicated.
How do they pair up?
The four involved chromosomes, the two normal ones and the two translocated ones, has to contort themselves into a cruciform or cross-like shape during synapses so that all the homologous regions can pair up.
A cross shape, okay? And how they separate determines the outcome.
Exactly. If they segregate properly in one specific way, alternate segregation, you get balanced gametes, either completely normal or carrying the balanced translocation. But if they segregate incorrectly, adjacent segregation, you get unbalanced gametes that have duplications of some segments and deletions of others.
Which leads to problems.
Right. These unbalanced gametes typically lead to non-viable offspring. So individuals carrying a balanced translocation often experience semi-sterility, literally, half sterility, meaning only about half of their progeny are likely to survive.
Is there a significant human example of translocation?
Yes, a very important one is related to Down syndrome, but it's a specific type called familial Down syndrome. This isn't caused by standard nondisjunction trisomy 21.
So what causes it?
It's caused by a specific type of translocation called a Robertsonian translocation. It typically involves the fusion of the long arms of two acrocentric chromosomes, where the centromere is very near one end. Most commonly, it's chromosome 14 and chromosome 21. The small short arms are lost, but they contain non-essential genes.
So someone can carry this fused chromosome.
Yes, a carrier of this 1421 translocation has only 45 chromosomes in total, because 14 and 21 are joined. But they have essentially all the important genetic material. They are phenotypically normal. They're balanced carriers.
But the problem comes when they make gametes.
Exactly. Because of how that fused chromosome and the normal 14 and 21 can segregate during meiosis, there's a significantly increased risk, much higher than for the general population, of producing a gamete that effectively has two copies of the crucial long arm of chromosome 21, one normal 21 plus the fused 1421.
Leading to trisomy 21 in the child if that gamete is fertilized.
Precisely. That's familial Down syndrome. It runs in families because the balanced translocation itself is heritable.
Wow, that's a direct link between a structural rearrangement and a major clinical condition. Okay, let's wrap up the structural changes with one more category. Fragile sites.
Right. Fragile sites are specific locations on chromosomes that, under certain laboratory conditions, like culturing cells in specific media, appear as gaps or constrictions and are more prone to breakage.
Are these common?
There are many fragile sites known across the human genome, but most don't seem to cause any problems. However, there's one major exception linked to a significant human disorder.
Which is?
Fragile X syndrome, FXS. It's associated with a fragile site on the long arm of the X chromosome, and it's the most common form of inherited intellectual disability.
What's the mechanism behind Fragile X? It's not just breakage, is it?
No, the fragility is more like a marker. The actual cause lies within a gene called FMR1 located at that fragile site. This gene contains a repeating sequence of 3 nucleotides, CGG.
A trinucleotide repeat.
Yes. In most people, the number of CGG repeats is small, maybe 6 to 54. But in individuals with Fragile X syndrome, this repeat region becomes massively expanded, sometimes containing hundreds or even thousands of repeats.
What does that expansion do?
When the number of repeats exceeds a certain threshold, around 230, it triggers a chemical modification process called methylation in that region of the gene. This methylation effectively shuts the FMR1 gene off, silencing it.
So no gene product is made.
Correct. The FMRP protein, which is crucial for normal brain development, isn't produced, and that leads to the symptoms of Fragile X.
And there's something about anticipation here too, right?
Yes, genetic anticipation. The CGG repeat tract is unstable. In individuals who are carriers, they have an intermediate number of repeats, maybe 55230, called a premutation. The repeat number can expand further when passed down to the next generation. This expansion tends to happen primarily when the premutation is passed through a female.
So the condition can become more severe or have an earlier onset in subsequent generations.
That's the general idea of anticipation, yes. And finally, just a quick note. While Fragile X is the most studied, there's also evidence linking some other fragile sites, particularly on autosomes, to cancer. Genes located near these sites, like the FHIT gene on fragile site, FRA3b, are often found to be deleted or altered in certain types of tumors, suggesting these regions might be generally more susceptible to breakage and mutation.
Okay, so stepping back from all these details on allopolyploidy, polyploidy, deletions, duplications, inversions, translocations, fragile sites, what's the big takeaway for you, the listener?
I think the main theme is just how critical genomic architecture and balance are. Phenotype isn't just about the sequence of A's, T's, C's, and G's. It's about having the right amount of genetic material in the right place and orientation.
These large-scale changes, these mutations, they're often really detrimental, sometimes lethal. But as we saw with duplication, sometimes these very errors can provide the raw material for evolution.
Exactly. They're not always just mistakes. Sometimes they're opportunities for innovation over evolutionary time.
So maybe a final thought to leave you with, drawing a bit on the ethical dimensions our source touches upon. We talked about Down syndrome, Fragile X, cri du chat, conditions caused by these large chromosomal changes. Today, we have increasingly sophisticated prenatal diagnostic tools like non-invasive prenatal testing and IPT using just maternal blood that can detect many aneuploidies very early.
Right. The technology is advancing rapidly.
How does this growing knowledge, this ability to see these architectural variations before birth, challenge or maybe reshape how we think about genetic normalcy, variation, and our responsibilities?
That's a really deep question. Knowing about these potential outcomes forces us to confront complex ethical considerations about screening, choice, and what we define as health or disability. It's something society is constantly grappling with as our genetic understanding grows.
Definitely something to ponder. It really connects the molecular details we discussed back to very human questions. Thank you so much for joining us on this deep dive into the world of chromosomal mutations.
Always a pleasure.
We'll see you next time on the Deep Dive.